Okay, so for this video, you are going to need, we're going to finish up these notes here that we, right underneath the multiple alleles with the blood type. And then we're going to do the first page of this note packet. So you need both of those for this. Okay, so let's look at some more details about ways certain traits are inherited. So multiple alleles were one of those. So we did the 3 different alleles, and everybody has a combination of 2 of those. One that you get from mom, one that you get from dad. Polygenic inheritance, poly means many, right? And so polygenic inheritance is when multiple or many genes, genic referring to genes, affect a single phenotype. So there's an additive effect of 2 or more genes on a single phenotype. An example is skin color. Most things that you have like over a wide variety of range, like skin color or height, short to very tall, like I said, skin color, very pale to very dark, are polygenic. And let me give you an example with the skin color. You have some room here, so we're going to write some things. So let's say the skin color gene, let's say there's 3 of them, all right? So let's say on chromosome pair number 4, so this is the DNA before it's replicated and so on. So on chromosome pair number 4, let's say there's 2 skin color genes, and let's say this person is a big A and a little a. All right, and then let's say there's another skin color gene on chromosome pair number 10. And let's say that person is big B, little b. All right, so they have 2 genes, one that they got from mom, one that they got from dad. And let's say there's a 3rd skin color gene. And let's say that's on chromosome pair number 15, and we'll use D's. Let's say they're big D, little d. All right, so they've got 6 total genes that determine what skin color they have. So how does this work? So in the skin color genes, let's go over here, the alleles. All right, the alleles for this, the dominant, means that they produce a pigment called melanin. Melanin is, whenever they have a dominant gene, that codes for making this pigment. This pigment then gets deposited in the skin, and the more melanin you have, the darker the skin. So therefore, the more dominant genes you have, the darker the skin. Recessive genes, so any of these recessives, there's no production of, so I'm going to put do not produce melanin. So for skin color here, we would say this particular person's genotype, they've got 6 total genes that are helping to determine the color of skin. So they've got the A gene, so they're big A, little a, and we just write it all together. They have the B genes, big B, little b, and the D genes, big D, little d. That's their genotype. So out of the 6 genes, this person, whoever it is, their phenotype, out of the 6 genes, they've got 3 not producing melanin and 3 producing melanin. So half of their genes are producing melanin, which means that they would be a medium shade in terms of skin color. So let's look here then. What would be the darkest shade? Well, the darkest would be making the most melanin, so therefore they'd have to have all their genes making melanin. So the darkest shade would be big A, big A, big B, big B, and big D, big D. And what would be the lightest? All recessives, right? That means they're not making any melanin. All right, that would be the lightest. So you can imagine, so there are 6 total genes here, 3 pairs. that I did in this example, imagine if there were 10 pairs or 15 or 20, how many different shades you could have. So that's more realistic. I just did 3 to illustrate a point, but like you can get so many different colors or shades, and same thing with height. So oftentimes genes that show are governed by polygenic inheritance or inherited that in that manner looks like a bell-shaped curve. If you look at the number of people on the y-axis, and you have all your shades from lightest all the way to the end darkest, right? What this looks like is a bell-shaped curve. What does that mean? Most people are in the center, and then as you go to the extremes, all recessive, there's very few people. And as you go to the extreme, all dominance, there's few people. All right, so polygenic inheritance in a population generally shows this bell-shaped curve. All right, so that's another yet unique instance of a different way that things are inherited. And the last thing on this packet that I wanted to talk about is sometimes the environment impacts the phenotype. So sometimes individuals can have the same genotype, but if they're put into different environments, their phenotype is the same. So this is called multifactorial genes. Both genetic and environmental factors influence the phenotype. So I'm going to give you an example with hydrangea flowers. Flowers show something called phenotypic plasticity. All right, that's a new word. What does that mean? I'll put it under here. Individuals with the same genotype exhibit different phenotypes in different environments. Okay, so hydrangea flowers, I will show you a picture in class, but hydrangea flowers are generally pink. And then if they are grown in acidic soil, so if you put, if their soil is more acidic, their flowers will be blue. So if you grow hydrangea flowers and you want those flowers to be blue, there's actually a product that you can buy in the store to sprinkle on the soil around the hydrangea flowers to make the soil more acidic, and the flower will be blue instead of pink. All right. Okay. So now we're going to move on and focus on this next handout right here, the chromosomal basis of inheritance. All right, so this, notice that this said Mendel's discoveries. Gregor Mendel, father of genetics, crossed the P's, came up with dominant recessive genes and all these things. Thomas Morgan, also is a famous scientist, and he worked with fruit flies. All right, so he mated fruit flies, and he came up with a couple of things adding to our genetic knowledge. So he was the first to associate a specific gene with a specific chromosome. So when we wrote over here that skin color, there's a skin color gene on chromosome pair number 4, he came up with that there's a gene for, not for this particular one, but he came up with the fact that a gene was found on a particular chromosome. All right. We talked about, I'll give you another example here when we did the pedigrees. Here we talked about cystic fibrosis, this respiratory disease. They know that that's on chromosome pair number 7. All right, so genes are found on specific chromosomes. All right, and then B says he used fruit flies. Fruit flies are pretty easy organisms to use. They have a 2-week life cycle, so you can get generations and generations of flies. You can have 2 parents mate, have a kid. That kid only grows and lives only 2 weeks, and while they're alive, you can have them mate, and then you can get lots of generations and lots of data very, very quickly with fruit flies. And they're pretty simple in terms of that they only have 4 pairs of chromosomes. Let me ask you this: how many total chromosomes do they have? They have 4 pairs. If you said 8, you'd be correct. All right, now to make it a little bit more confusing, he used some different terminology here that we need to be familiar with. There's common terminology. He used this word wild type. All right, so wild type. So this is one word we need to know. It says normal phenotype for a trait. This is what's most common. All right, so he used, which I think can be confusing, he talked about what's most common versus a mutation or what's rare. So a common one for fruit flies that he looked at is their color of eyes. So the most common color for a fruit fly's eyes is red eyes. So red eyes is the wild type because it's most common. So if you ever see fruit flies, you leave some fruit out and some flies are, you can actually, if you look real closely, if they sit still for a minute, you can see that they have red eyes. Then there was the opposite, the mutant phenotype. He called anything that wasn't a mutation or wasn't a wild type a mutation. So these are traits that are alternatives to the wild type. All right. So example is an alternative color is white eyes. Okay. So example here, red versus white, all right, eye color here. So his terminology, which I have to say I don't love, he used W's. He used the W because he goes with the mutant phenotype. What, by the way, wild type and mutant, let's go back for a second. A gene, let's say this is the piece of DNA. All right, and you have 2 sides of DNA. Remember, you have A's on one side, that hydrogen bonded T's on the other side, and it's the order of these bases that determine what protein is going to be made. So let's just say there's C, C, T, A, C for this side. So what would be the other side? This would be G, G, A, T, G. And let's say, I'm just going to make it from right here to right here is a gene, an eye color gene. So when we say, like when we did over here, that I said the skin color gene is right here, that's what I'm doing. I'm saying there's the little piece of DNA with the order of the bases that code, like reading a book, code for making a protein. And so the wild type gene here is coding for making a protein that is red to give the individual red eyes. So most flies have this gene that code for making the red protein, which gets deposited in their eyes and gives them red eyes. The mutant phenotype, what he's saying is that anything that deviates from that wild type or most common, something along the line happened in one fly's gene that mutated it, and instead of making a red protein, they make a white protein. So the wild type is the most common, and that's why he calls it a mutant phenotype. He's saying that there was the wild type gene got mutated, changed an A to a T or something like that, codes for making a different protein, and all of a sudden you get a different phenotype, white eyes. All right, so how he denoted this, and this is the part that I don't like, he took the mutant phenotype, which in this case here is white eyes, so that's why he used a W, and so the white eyes is W, and then the red eyes is the wild type, and so he uses a plus for wild type. So the plus here is really important. That means that it's wild type. Now notice here is that it shows, I have here written that the white eyes actually happens to be recessive and the red eyes happens to be dominant. That doesn't always have to be the case. Sometimes the wild type, the most common, is not the dominant trait, all right? Like I used the example of polydactyly with humans having 6 fingers. The wild type in humans would be having 5 fingers, but that is recessive. Having 6 fingers is actually a dominant gene. So it doesn't always work out that way, that the wild type is dominant. It happens to be this case in the eye color. So when we do this, I like to show, like if you're going to use a W like this, I use a little W, and then I make it really a big W with the plus, because what that tells me is if I use this denotation, it's telling me what's wild type, and it's also telling me that the wild type is dominant to the mutant. All right, so that's how I use that. He did not, like I said, which I think is confusing, and I think it's been confusing for students in the past. All right, and so another example is curly versus straight wings. So notice here, I use this to show that, to bring that point home. Curly is the mutant, and notice he used a symbol representing the mutant gene, which is curly, so he used CY. All right, these letters are just symbols, so you don't have to write out the gene. And the CY plus is the wild type. All right, so there's the plus for the wild type. But notice here the wild type's recessive. Straight wings is recessive, and this is dominant. So therefore you could do, since this is dominant, we could do a CY, and then here a CY like this with the plus. So dominant, recessive, so it doesn't get confusing there. Okay, so somebody who, a fly who has straight wings, what would be their genotype? Straight wings. What would be their genotype? Pause it if you need to. If you said to be straight is recessive, so it'd be CY plus, CY plus. And let's just I'm just going to put this out here, that the plus is the wild type, not necessarily dominant or recessive. Okay, so that's some of his terminology that he used. Now, he also, since he linked genes to particular chromosomes, he also noticed that the sex chromosomes, the X and the Y, XX if you're male, or sorry, female, XY if you're male, that there are genes on those. All right, so he called those sex-linked genes, genes for a trait that are located on a sex chromosome carried on the X or the Y. Most of them, I want to say to you, is the X chromosome, because when you look at the X chromosome is really big, the Y chromosome is really small. There's just simply more DNA on the X chromosome. This is why if you don't have any X chromosomes, you cannot survive, but you can survive without a Y chromosome. There's just less genes on there. The genes that are on Y chromosomes are the genes that determine maleness. There's a particular gene called the SRY gene that produces codes for making an embryo as they're developing into a male versus a female. So if you do not have that SRY gene, the default sex is a female. All right, so you have to have that Y chromosome with an SRY gene to develop maleness. All right, so let's look at on the X chromosome. So since X chromosome is so big, so much more DNA, most sex-linked genes are on there. So on the X chromosome, let's do a trait. Let's say the trait, let's do a real human trait on the X chromosome, is color blindness. More specifically, red-green color blindness. All right, I'm going to just use CB for color blindness here to do dominant and recessive. So here, what we do for color blindness is since it's on the X chromosome, and since females have 2 X chromosomes, so you have 2 color blindness genes, but males only have 1, because males have X and a Y. So therefore, they're only going to have one colorblind gene. So what we do is we denote that it's on the X chromosome. So we do an X, big X, with a superscript. I'll use A's for colorblindness here. I'm going to switch it up here. That dominant is not colorblind, so I'm just going to use CB. Not colorblind is dominant. If you have an X with a little a, that means that's the colorblind gene. So let's look at this. If you're female and you're heterozygous for the colorblind gene, what genotype are you going to be? Now, to really learn this, I would pause this and actually think about it, all right? So female has 2 X's. So you have to say, okay, what's going to be on her 2 X's? Well, if she's heterozygous, she has to have a big A on one X and a little a on the other X. You're crossing them with a colorblind male, all right? Males have 1 X and 1 Y. He's colorblind. There is no gene for colorblindness on the Y chromosome. The X and the Y chromosome have different DNA. They are not homologous chromosomes. So there's only a colorblind gene on the X. So to be colorblind, he has to have a little a on his 1 X chromosome. So even though the colorblind gene is only on the X chromosome, we still put the match, the pair, I should say, with the Y there for his 2 sex chromosomes. Another thing I'm going to introduce to you is some symbols. The universal symbol for a female is a circle, the little plus coming down here. In a male, the universal symbol, the circle has an arrow going up to the upper right. Okay, so let's do a cross here. Okay, so let's do the mom along the top. She's going to pass on one of her 2 X chromosomes. So she could pass on the X with the big A and the X with the little a. And then dad, what could he pass for this chromosome? An X, and his X has a little a. Or he could pass on the Y. So he literally makes half of his sperm cells with an X and half of his sperm cells with a Y. All right, so then this sperm fertilizes this egg cell. You get a kid with an X big A and an X little a. This one, X little a, X little a. This one, X big A, Y. And then X little a, Y. Notice that we always put the X's first. That's just the way it is. All right, now let's do the phenotype ratio. I'm not going to have us do the genotype. Oops, I can't write and talk at the same time. Phenotype ratio. Phenotype is their physical appearance. With sex-linked traits, what we do is we also separate males and females. All right, so what do we have? So let's do the females first. So right here in this box, is she colorblind? It's a girl. She's 2 X's. So we have a 1, and then she's not colorblind. So I'm just going to put not CB, and she's a female, so I'm just going to use my symbol. All right. This is why the symbols become important. All right, then colon. Then let's look at the next one. They could have a 1 colorblind female. Colon. See if you can do the rest of it. I would pause it and see if you can do the rest of it. I'm going to go ahead and do it. Hopefully you paused it. You should have 1 not colorblind male. And then lastly, 1 colorblind male. So what does this mean? Every time they have a kid, they have a 1 out of 4 chance of having a girl that's not colorblind, 1 out of 4 chance of having a colorblind girl, 1 out of 4 chance of not having a colorblind boy, having a not colorblind boy, and 1 4 chance of having a colorblind boy. All right, so that's that. So you can see they still have a 50-50 shot of having a girl versus a boy. Let's say they have a baby girl. All right. So now if they have a baby girl, you know it's a girl. What chance? You probably don't know when they're born right away. What chance is that baby girl going to be colorblind? 50%. All right. Because you already know she's a girl. All right. So that's kind of how you analyze that. All right. What I'm going to have you do is pause this and do this problem all by yourself, just like as we would in class. All right. Do it even though I'm not watching. All right. So pause it. Okay, so hopefully you came up with that. The homozygous recessive female, she's got her 2 X's. Homozygous, little a, so she has 2 little A's on her X chromosome. Normal male, he has an X and a Y. Normal, not colorblind gene is a big A. So that's your cross. So then you look like this. All right. X little a, X little a. X big A, Y, right, X big A, X little a, X big A, X little a, X little a, Y, and X little a, Y. All right, so now the phenotype ratio. Again, if you did not pause it earlier, why don't you pause it right now and do this? It would be 2 not colorblind, and then these 2 are female, so the symbol for female is this, to 2 colorblind males. All right, and so look at this. All right, all the boys are colorblind, and the girls not colorblind. Color blindness and things on the X chromosome happen more often in boys. Why? Because males only have 1 X chromosome. So look at mom gave a little a, an X with a little a, to both her daughters and her sons, but only the sons have color blindness. Why? Because females have a second X chromosome that they inherit from their dad that has a possibility of covering up that color blindness gene. Males, on the other hand, inherit the X with the colorblind gene, and boom, they have it, because they don't ever get another X. They don't get another X to mask it. So therefore, they get the X with the recessive gene. That's what they get. So most often, colorblind people are males. All right. So I'm going to have you do this packet here that you picked up. So sex-linked inheritance problem set. So these are